Good morning, everybody. Today we are going to start a topic called as electron transport chain. Now, when we talk about electron transport chain, it is a topic from the chapter respiration, and we have this to be done after citric acid cycle. So, in citric acid cycle, we have seen that the reaction happens, and then there is another product formed, and then there is another product that is formed based upon the enzymes which are active. In here, the similar thing happens, yet we just want to know that the electron which is present at this end travels to the other end. How does this happen? It happens with the help of proteins. And before that, we will see where exactly it happens. Let us have a look. As you can see over here, I have drawn a small little mitochondria. So basically, the mitochondria is the one which actually takes care of this electron transport chain, which is the energy currency of the cell. Now, there is an outer membrane and then there is an inner membrane. At the inner membrane, you have these projections called as cristae. And these proteins which are there, or this complex is the drawing that you can see, it is just an enlarged image of this small section out here. Okay, just let us have a look as to what are the requirements to understand an electron transport chain. First, we should know its location, that is in the mitochondria. Okay, and at the same time, it is on the lipid layer. Now, the lipid layer has proteins. We call them as integral proteins, and also we call them as peripheral proteins, which is a little bit hanging on the outside. Now, we have to consider and know the location that this area above this drawing is your intermembranous space. Okay? Intermembranous space. And at the bottom over here, this place is the matrix. Fine? We are supposed to be also knowing that these are proteins, and presently for your class, it is only enough to know that they are powered by complex 1, which is this one, the first one, complex 2, that is with this one over here, and then we have complex number 3, which is this one. Complex number 4 comes at the last. And at the same time, we need to be considering about a few more molecules, which is the ubiquinol, cytochrome P and cytochrome C. Cytochromes, they are electron carriers. Fine. Ubiquinol is also electron carrier. But then these complexes which are there, they are the proteins. Now, talking about the ubiquinol. Now, this is a special molecule which goes to and from complex 1 to complex 3. Now, let us see how it works. So far, we have understood that this setup is required to take an electron from this end to the other end. Now, we need to also see that there is one more thing, that is what is the reason that this electron has to move? It has to donate oxygen. So the oxygen has to be donated at this end. And at the same time, electron transfer has to take place. And the electron transfer helps in the building of ATP. Well, this is presently not under this topic. The next topic is called as chemosynthesis. Okay, so that tells you how ATP is usually made. First, let us understand this, and then we will be switching over to the next topic, which is going to be chemoosmosis, which will help us to understand how ATP is actually produced. So far, we'll just know the functioning of this. Fine. Now, at this end, at complex number 1, electron is given in. Okay? Now, electron is given in with the help of NADH, that is nicotinamide diamine. amine. Okay, this is the hydrogenated form. Now it will lose one electron and it will turn out to be a proton. And that's why we have this electron which has been taken from this hydrogen and which is leaving it as a proton, and it will travel up complex number 1. Complex number 1 will take it further into its further regions, and again it will uptake another one. At the same time, you have this electron traveling to ubiquinol. The ubiquinol will take 2 electrons at one time, and then it will travel towards your complex number 3. Complex number 3, 1 electron, it will be given to cytochrome B, and there is another one called as cytochrome B2. Fine? So it will take it. Now what's happening at this end is the redox potential of complex 3 is very high. So it takes away the electron. That is why it is taking away the electron very fast. And so this one gets devoid of a particular electron, and then it comes back to